I hope that you've all had a very enjoyable lunch. I've had way too much to eat, so if I fall asleep, then it's because I didn't manage to get my coffee, but then I was sort of chivied along. But first of all, disclaimer, of course I believe in follow-up in patients with uveal melanoma, but I do feel it is important that occasionally we stand back and reflect on our own position, because in Southampton we now offer everyone interval scanning, and I think it's very useful to occasionally take stock and think about whether we're actually doing the right thing. But when I saw Wendy's horrified face when I stood up and spoke against follow-up and follow-up with the MRI in particular, I thought I'd want to clarify that just for the record. Okay, so immune checkpoint. So the big change in oncology globally has happened in the last 5 years, is that the cancer attack through the immune system is becoming almost a byword for improvement and change. And so I thought I would take you through a bit of how we think about this, and then talk to you about some of the UK data that exists with these drugs. And ultimately, this is going to be a personal perspective on the human immune system. And you have to bear in mind a reflection of how I tick, because I sort of get up in the morning and think about how might you want to understand and then manipulate the human immune system. So my enthusiasm for this field is clearly reflected in how I speak about that. So this is what we are ultimately after. So we see the tumor cells sitting in the background here, the big flat shape, and these little round balls. That's the type of cell, these effector cells, T cells, cells that come out of the T thymus. So a gland behind the breastbones where our T cells go to school. So this is the education that all our T cells get, and then they leave the thymus, but they retain the T in their name. And then they sort of patrol the body, a little bit like sniffer dogs, and they look for things that are foreign. And our immune system has not really evolved to fight cancer, but has evolved to fight infection. So all the stuff that is foreign to us, so when you've got the measles or the flu or the chickenpox, those are molecules that alter the way the cells in our body behave. And these altered signals are what our immune system is trying to recognize. And then if it does do so, then it becomes all activated and gets all het up, and then it settles around cells that are, in this case, infected or tumorous, and ultimately makes holes in them. And the molecule that's actually doing that is called perforin, so it perforates the cell membrane, and that's the way the cells explode, like Pippa had explained and demonstrated in her slide. Now, in normal circumstances, an immune system becomes really activated, and then when you've got the flu, you feel really lousy. So you've got the shivers, and the hair is painful, and everything's joints ache, and you have a temperature, and that's all related to immune attack. So the immune cells that are fighting the virus-infected cells release molecules that makes cell move, and these molecules that make cells move are called cytokines, translation for cell movements. And so normally, once this event has happened, once you've had the flu, then everything settles down again, the whole system calms down. And so it's in a constant attempt to reset its balance. So that's what's the problem in patients with cancer. So the cancers are trying to hide from the immune system. So they release molecules that switch the immune system off. And we're trying to achieve immune control to switch the immune system back on. And if that is unsuccessful, then the net effect is that the cancer grows, the lumps get bigger, the cancer in the liver starts waking up. And if you are successful, then ultimately the cancer disappears. And then we think that we can no longer see the cancer. And over time, when that goes on and on and on and on, then at some point, even the oncologists start banding around the word cure. And for cancer immunotherapy, that is a term that is sort of very cautiously raising its head in many clinical systems. And, I've been in this field for the last 20 or so years, and I've seen things come and go in melanoma land. But now we have drugs that actually people have been treated with, and 10 years on, they haven't had anything else other than 4 doses, in this case of ipilimumab, and the cancer still hasn't come back. So this is a landmark shift in how we think about cancer treatment. Rather than a bit of selumetinib and poison it, cancer lumps get a little bit better and then they come right back. So the remit to all of us, the oncologists, but also to the patients, is what can we do to try and make these sort of processes work better? So let's think about the players in this process. And some of it's important because when Pippa talked about the IMC GP100 study, that's one of the reasons why you need to know about this. So here's the cell in either your tumor cell or your infected cell. They are called, in this case, I've labeled it as a genetic cell. So these are the cells that the sniffer dogs, that's the guys up there, try and sample. So they have this molecule on their surface that you can imagine that like a nose, and they patrol our tissue all the time, so your lung and your skin and your bowel and your liver, and they look for things that they might be able to recognize. And then they do recognize these little bits of proteins. So that can be a protein, a chopped up bit of a virus or a chopped up bit of a tumor. And that is sort of like in a chalice presented to the outside world from the antigen or tumor presenting cell in this molecule that's called the HLA molecule. Now, if you want to target peptides, this bit, from a tumor, then you need this molecule to be of the right flavor. Because if this is the wrong flavor, the wrong type, then this molecule can't see this. So if the vaccine that you're making or your GP100 molecule targeting T-cell receptor will only see this peptide, it is presented in the HLA-A2 molecule, it's useless to give this stuff to people who don't have this HLA-A2 molecule because the T-cells can't see it. So this underlies a very specific interaction. It's like the fingerprint of our immune system. And this fingerprint is determined by these HLA molecules, and there are two flavors, the MHC class 1 or HLA class 1 and HLA class 2. HLA stands for human leukocyte antigen, so the human white cell antigen, so the molecular fingerprint. Now, your peptide is now presented, and that's demonstrated here, and then the T cell comes along, sniffs this out, and then binds with this T cell receptor to the peptide in the tumor. But that's really only. Okay, so that's the critical interaction that needs to happen in order for the immune cells to be able to do anything useful. And there, I've labeled two flavors of immune cells. So these are the affected tumor cells. They're the guys with the teeth. So the dogs, the sniffer dogs that can actually take out the cancer cells by, for example, releasing these perforin molecules. And these guys are almost like a support group. They facilitate the CD8 cells to be activated, and they help the T cells to do their job. So they release cytokines, molecules that make the effector cells, the dogs, actually be able to do this. So you can imagine the CD4 cells a little bit like the supply troop. So without one or the other, nothing happens. So an immune system needs both of those. And what's becoming very clear is that many of the vaccines that we've used in cancers that were exclusively focusing on this actually fail because we didn't take these cells into account. So again, as always in human biology, things aren't quite as simple as they seem. And of course, I'm abstracting this because it makes it visible. So here's an example of such a tumor, and I do apologize that my slide isn't quite bright enough, but here are the red cells that pick up, the red color that picks up the tumor cells in a blue sea. So this is the cell nucleus, the blue is the cytoplasm, and in between are spaces. These spaces are filled with other stuff. So other stuff being proteins, cells, fibroblasts, cells that sort of confer the structural integrity to the T cell, to this tumor, and also to our liver or our lung. Without those, the cells that are actually doing the functioning would all fall apart. So this is a sort of, the green stuff is the glue that holds the tissue together. But if you're a T cell, and now you need to use your imagination, you can possibly see that there are these red cells. I've picked out the cytotoxic T cells. They are sitting in this mess here in the middle, but they're not actually going to the tumor. And what's happened is that the stromal cells have made a molecule that's called the PD ligand 1 molecule that switches the T cells off. So what I'm saying is, yes, you need that interaction between the tumor and the T cells, but you also need to overcome additional hurdles for the T cells, the immune cells, to actually make it to where they're supposed to go. And if this is PD ligand 1 positive in the stroma, then you would have thought that a drug like pembrolizumab or nivolumab, an anti-PD-1 antibody, would be a really smart move for this patient. So what we've now spoken about is the specificity of this interaction. And for some of the new drugs that are being developed, this is really an exquisitive specificity. So unlike Ipi or Pembro, you give it in, you drop it in, make everything crank up and hope that it will work because it will find its own way. For something like the IMC GP100, you only adding sort of this one component, so that's why it needs to be in patients only with the HLA-2 molecule. But if you have only this interaction or only this interaction, then the T cells ultimately go to sleep. That's called a process of tolerization. So it's a way in which our immune system is switched off. So it needs a second signal. This is the first signal. It needs a second signal to make the whole process run. So the second signal is determined again by one partner molecule on the tumor cell and one partner molecule on the T cell. And these are called because they're also needed, stimulatory because they activate molecules. So this second signal is what the whole fuss is about in oncology land, because the moment that happens, the cells then start making lots of cytokines. So, for example, I've written the IL-12, interleukin-12, which is one of the key cytokines that makes these processes run. And at the top is interleukin-2, IL-2. And you may have come across IL-2 also as a pharmacological treatment. So give lots of what makes T cells work in the hope that by using this as a blunderbuster approach, you might just get away with pushing the right cells in the right direction. But for the purpose of this talk, this is the synapse that we really need to think about. That's the synapse we need to think about. And so targeting these has really been quite a dramatic thing in oncology. And basically what we have available now are antibodies, synthetic molecules that block this interaction. So you give a lot into the blood, it goes wherever the T cells are trying to see the tumor cells, and then they stick on this part of the antigen. So they stick in between these two molecules. But in order to understand what happens, you need to know one other thing, and that is that these molecules come in multiple flavors again. So the first interaction switches the T cells on. But the moment the T cells are switched on, and it is really bad for T cells to run away, it makes you really ill, not just like having the flu, but all these immune toxicities are related to not switching them off again. So the moment the T cells are engaging, they also upregulate the off switches, and they look just like that. So the off switch here, this is the on switch in yellow, would be replaced by an off switch that has just of a different flavor. And this binds to exactly the same molecule, but the function for the T cell is quite different. So where the first interaction is needed to make the T cells run, the second is needed to make the T cells stop running. So the net result is you have an activated T cell, it clears the virus, and then the whole system calms down again. Now, it turns out that cancers are really smart. They, not in an intellectual sense, but probably just by selection, they make molecules that do just that. So if you start looking in the tumor cells, and in the tumors, and look at the T cells, they all have this phenotype, this characteristic of being exhausted. In other words, they have tried seeing the tumor, because that's how they got there in the first place. But then the tumor has switched off the T cells. And this is a process called exhaustion, just like many doctors and many patients. And that is a real problem, because these T cells are now just sitting there and not doing anything. And so with the new drugs, with ipilimumab and pembrolizumab, what you try and do is interrupt this synapse. So this interaction that switches the T cells off with the idea that if you take the brakes off again, then the whole system will start cranking up. And indeed, that's what happens. So if you give this to people with melanoma or with lung cancer, then all of the immune reactivities become activated. So the problem then is you're not just activating against the cancer, but also against all the other things that are normally switched off. So suddenly T cells that see your skin or your brain or your heart or your kidney or your liver also run amok. And that's where the problems come from, the immune-related toxicities. But there's one other bit here that is really critical. If you want to release a T cell, it's got to be there. So if you want to make a T cell that has been switched off work better, you have to have a T cell that's already there and that's seeing the antigen. If that's not the case, then the whole system doesn't work because there's nothing to release. It's like a car that's sitting on the flat. Well, you can take the brakes off all you like, it won't run because there's not a hill to run down. So I think what we'll learn in the coming years is that the amount of T cells, or the number rather, of T cells in the tumor give you a flavor of how likely it is that these drugs will work. And I think it will turn out that actually in uveal melanoma there aren't very many of these. And the first data that we've looked at suggests that in the liver that is also the case. But that might explain why these drugs don't work so well for uveal melanoma, and that actually what we need to do is something else that makes the tumor more appetizing and attract immune cells. And I thought that the story that Peter Schlosserek presented earlier on starving the tumor cells and making them more visible in that way would be a really smart combination with an immunotherapeutic, and I'm sure he's thinking along those lines. So then what is it with the drugs that we currently have? Well, this is data from the UK. This started in 2011 from when BMS first made ipilimumab available. And Pippa was the senior author on the publication that presented these data. This is from the Expanded Access Program that was roughly 200 patients in the UK. And we looked at whether the 20% or so of patients who were treated with uveal melanoma looked any different to patients with skin melanoma. So what we're graphing here is time at the bottom, and then at the top you start with 100%, everyone's alive. And then as time goes on, each step in the curve means that one patient has either progressed or has died. So what you can see is that this curve has much finer little steps. That's just because the numbers of the patients are much, much bigger. And this has coarser steps because there are many fewer patients with uveal melanoma that have been treated. But it's actually quite surprising. So it looks as though the curves are really quite similar. And it looks as though the long-term outcome here, measured at least to these, so it goes up to 40 months, that's, well, just under 5 years, 3 and a bit years. So that actually the overall survival in this very small cohort compared to a slightly larger cohort is relatively similar. I found that quite surprising. So when we first started looking at these data, we also went back and had a look at what happened in another country. So this is a similar cohort from Italy. They've also had an expanded access program, and a chap called Michele Maio from Milan and Paola Certo from Siena put these data together and published on 80 patients with uveal melanoma. And actually, the curves are relatively similar. So this doesn't look that much different from what we're used to with cutaneous melanoma. And if you map them onto each other, then this is the data that comes out of the registration trial, so the data that is from the large randomized phase 3 study in patients with skin melanoma, and here is the data for uveal melanoma underneath. So it's not quite as good, but actually quite comparable between the countries, and it suggests that although this drug is not staggering, it does seem to make a difference to some patients if we could just pick those out. And similar data are now beginning to emerge for pembrolizumab. And I haven't got a comparator data set because we haven't been able to map it onto the randomized data sets yet because those aren't available. But this is our own experience in terms of overall survival and progression-free survival of the first 20 or so patients that we've treated with pembrolizumab in Southampton. And again, it looks as though there is a group of patients who do really rather well long term. So this is days. You can see it's much shorter follow-up. It's only just about a year. And it looks as though this drug also has some activity. What we found really quite staggering, though, is how big the difference is according to where the cancer is. So if you have extrahepatic progression at the time you get the drug, so cancer that is growing in the skin or the lung or in other bits of the body, and that's not selected for any particular subgroup because the numbers are too small, as opposed to having disease progression in the liver, you can see that patients whose liver disease is not controlled do much worse. And that's even in this tiny data set highly significant. So what we think is going on is that the outside of the liver, both ipilimumab and pembrolizumab have some activity, but it isn't as staggering as in other cancers. But if we could just tweak the system a little bit better, we might be able to control things better. But what we do need to do is get control of what's going on in the liver. And these data are sort of shaping the way we, or I think, about the management of patients with advanced uveal melanoma. Because in the past, we've gone about, we've done scans, and then when there was liver-only disease, we would have thought about treating patients with liver-only disease. But maybe these data suggest that we should reflect again. And maybe what we should be doing is focusing on controlling the liver disease as the first port of call, but not being worried only by what's going on in the liver, because we could then perhaps include other treatments such as ipilimumab or pembrolizumab, much along the multimodality approach that Arjun had presented earlier on. So provocative data, and clearly, a tiny data set if you think about how many patients are needed for many of the big trials, but it suggests that we need to also start reflecting on how we manage liver disease in the context of disease that's not only in the liver. And perhaps we should be much more aggressive in managing patients who have wider disease than we had initially thought. Clearly, there are many other molecules. So if we think about combinations, then in skin melanoma again, and now in lung cancer, the first studies have reported or are ongoing with the combination of the two drugs. That's going to be difficult because it makes the side effects a lot bigger, but in at least skin melanoma makes the response rates much better. And then there is a whole bunch of other drugs that are sort of coming up in the background. So CTLA-4 and PD-1 as targets, so ipilimumab, and the other compound is called tremelimumab. And then anti-PD-1, which is called nivolumab, and pembrolizumab. MAB stands for monoclonal antibody, so it's a synthetic molecule that makes a lookalike of what our body would normally make in soluble form. But these are the molecules that are coming up really quickly. So LAG-3 is already in clinical testing. I seem to have lost my TIM-3. Oh, it's the third one under here. So TIM-3 up there. And then activating molecules, so OX40, GIT receptor, CD137. So I think there'll be lots of new compounds that may tweak the immune system slightly differently according to the context that things are happening in. And then I was going to speak a little bit more about co-stimulation targeting HLA-A2 molecules, but Pippa's already shown the data, so I'll just skip that, other than saying that I'm really very excited about this novel concept. And actually, in terms of answering one of the questions, could this only be possible for patients with HLA-A2? Of course, in principle, it would be perfectly reasonable to make these same IMCORs against molecules that are HLA-A1 or HLA-A3, but you'd need to persuade, or we'd need to persuade the company to do that. But technically, that shouldn't be a challenge. So if this is what we're trying to get at, then what can you learn about the tumor cells and the interactions and the immune cells? And try and make smarter trials. And so our particular flavor on this is to really take the tumor apart. So here's your mashed up tumor, which you've sort of fed with molecules that have eaten all the stroma away and have left the tumor cells and the immune cells, and then to sort them into little pots, like bead collecting. So maybe cytotoxic T cells here, your tumor cells here, and the green might be your helper T cells. And we can do that now routinely. Certainly, we've done that for lung and head and neck cancer. We're just starting on melanoma samples. And then you can start asking about how similar they are. So we've taken here and compared in two different colors cells that come out of non-cancerous lung and cancerous lung. So lung cancer and cells that are sitting in the non-involved lung. So if you will call these tumor-infiltrating lymphocytes TILs, and these would be LILs, lung-infiltrating lymphocytes. And so the first story that comes out for us is really quite extraordinary. So out of these 20 patients, the tumor cells, or the TILs, the tumor-infiltrating lymphocytes, look really very different to the lymphocytes in the non-tumor, in the LILs. And what then turns out is that you can start reading out, and apologies again, it doesn't really project very well, but these gray ones are the ones that are negative, and the red ones are the ones that are positive. Then and then on the right-hand side, you can see the ones that come out of the tumor as opposed to the normal lung. And what you can see, probably, I hope at least, is that there is a group of little cluster of these tumor TILs that are all very rich in this PD-1 molecule. And if you then follow them and look for expression of other inhibitory molecules, they also make TIM-3, which is another of these inhibitory molecules, LAG-3, 4-1BB, and CTLA-4. So what it says is that if you look at the T cells, the immune cells in the tumor, not only can you start unraveling how they behave differently in the tumor to in the non-tumor, so for example in the blood. So the first message is you need to look in the tumor. But the second is that these T cells are often switch off at multiple levels. So if you were to just give these cells CTLA-4, which is in some of these is highly upregulated, it probably wouldn't get over the problem, because you would also need to look at something that blocks PD-1. And additionally, what it also says is that if PD-1 isn't activated on these cells, releasing that axis ain't going to work. So I think what we are learning is that as we are becoming more savvy at looking in the tissue and looking at these multiple aspects, we'll be able to develop tools that allow you to read out which drug you should use. So, for example, for this patient here, I think a combination of anti-PD-1 with an anti-CTLA-4 might be a really good idea. But if these molecules aren't switched on the T cells, you probably won't be able to release them. So I think that adds another level of complexity, but at least from a technological perspective, one that we can start unraveling. So I think we need to learn more about the lifestyle of tumor cells, but specifically uveal melanoma cells. So what is it that makes these cells go to the liver so exclusively and pretty much in every case, with a few rare exceptions? And what is it that makes them more resistant to immune attack? And I think this means diagnostic biopsies. So for the patients, it means considering whether you really want to have someone stick a needle in your eye. That is important, as we've discussed earlier, because it's all about working out what you might want to do with the rest of your life and knowing whether there is actually a concern. But I think it's also important because only with this kind of material will we finally be able to work out what the hell's going on and what buttons we need to try and push. I think we also need to know the molecular mechanisms that happen in metastatic disease, and we are very focused on rebiopsying everyone who comes to our center because we so passionately believe that this material is so critical. But of course, we also don't want to give them Delcath if we're not sure that they actually have metastatic uveal melanoma. But the associated question really is, what can you learn from this tissue? And for me, in order to work out what is happening with the immune cells. But for Peter, that would be working out what's happening with metabolism. And clearly, we need to then build better trials to do these, and that means working with industry, but it means working with the patients, which is, of course, why I'm here today. Thank you very much. I'm happy to take any questions, unless I've stunned you into silence. You seem to be all awake still, at least. That's something. Okay, well, thank you very much for your patience then. Oh, Leslie.
You could have a question.
I know, I was hoping that you would dig me out of the hole.
So, I was very surprised by the data that you showed on the ipilimumab on the patient access, because a lot of those patients were very, very advanced, so we were poorly selecting patients for treatment. So, if with careful selection, you might even get better figures.
With early disease, you might get better fixes.
Yeah.
And there's this whole question that we've also discussed about whether radiotherapy would make immunotherapy work better. And there's very intriguing data that's emerging outside of the liver, where you give radiotherapy for lung cancer and give immunotherapy with it. And we have a quite substantial body of experience now in patients with uveal and other melanomas, where we have been very able to deliver safely radiotherapy with immunotherapy. So I think there's a whole question that needs to be asked around Certex as a localized way of delivering radiotherapy together with immunotherapy. And if I could find anyone who would be willing to give us access to the drugs, that's the trial I would really like to do.
Yes.
But of course, it also raises another question, because if this stuff is real, so if there is truly a durable benefit rate from these compounds, surely we should be using these in the adjuvant setting. So surely we should give this not when the horses have bolted, but when actually you can't see the melanoma. And I don't really buy the question of the trial size, because we know that there are patients who have a 50% chance of recurring in the next 10 years. So you can work out how many patients that would need to be. And yes, it probably would need to be several hundred, and it would probably need to be an international study, but I think you could answer these questions.
Yes.
And I think what's holding this at the moment is that the same questions in skin melanoma are still waiting for an answer. And the adjuvant role of ipilimumab has been answered positively, it works, but there wasn't a survival endpoint. So it was measuring time to cancer coming back, and that was significantly different. And of course, that's a reflection of the fact that everybody later on will of course get IPI, or PD-1, or both, as matters may be. So it becomes then much more difficult to have a survival endpoint. So I think that this is provocative enough in my head that I would want to see such trials being developed. and it would be really quite good to do that with, you know, with Pipocori and the melanoma group. But, you know, it would be really interesting to have your opinion of what would you want to be part of such a trial.
I mean, to me, whenever you talk to people, they always say, oh, ipilimumab doesn't work in uveal melanoma, but that's not actually what you've said.
I agree, and I don't think it is what is happening. So I think what is happening is it doesn't work well in the liver.
Yeah.
But then nothing does in the liver. So I think this is about working out what's different about the liver in the first place. And I think also working out whether the metastases that the patients have are truly the same in the skin or in the liver. And my gut feeling, based on no data whatsoever, is that they probably aren't.
Thank you.
Very good. Thank you for your saving question, Grace.
Does anyone else have any more questions?
Okay. Thank you very much.
Thank you.